All right, everybody, welcome back. We're going to start to delve into the DNA section of the course now, and we're going to take a different approach for these set of lectures, as we're going to focus on mostly the original research papers that delved into experiments that were specific for DNA replication, repair, and gene editing. So there are a couple of readings relevant to today's topic for DNA replication. There's first basic background information that can be found in the text in Garrett, and in particular for next week. And after we've gone through today's lecture, I'd like you to read this article, which is also available in Quirkus, so the discovery of DNA polymerase. And there are also some study questions that I posted on Quirkus, which I'd like you to use as a guide for your readings. And I think paying attention to the study questions and trying to glean the answers to those from the reading, then you'll really hit the points in the reading that I would like you to emphasize. All right, so I'm just going to take a few minutes before we start delving into the lecture and just offer up some few points as to how you can actually succeed in not only this section of the course, which is a bit more complicated, but also for the course in general. Usually we typically say, coming to class is very beneficial to facilitate your learning. And of course, since we're posting the lectures asynchronously, really devoting that 2 to 3 hours a week per lecture content to really focus and take in the material so that you learn the foundational concepts. Do take advantage again of my synchronous office hours, and definitely come to those and bring your questions as well. Really just try to spend your time very effectively as you're going through those lectures. And I think one thing that really develops over time is being able to take great notes. And taking notes really helps you consolidate your learning and understanding of the material, and really provides the context for more sort of complex scenarios. And in the next slide, I'll actually show you an article that I highly recommend reading on how to take notes. And of course, we're all scientists here. Obviously, do the readings. I do explicitly state which ones are required, and as such, since they are required, they are testable. I've also posted for these set of lectures a zip folder. You probably noticed as well, and it contains most of the original research papers that the various concepts and experiments that we'll be discussing are based upon. So those are meant as a resource. So if you want to look into them, you can extend your knowledge further by reading any of the papers in the zip folders. Another way really is to stay active in the course. Even though the lectures are asynchronous, there's lots of different ways that you can ask questions. Obviously, this is a big class, but, definitely coming to office hours, you'll have an opportunity to ask me any questions that you might have, whether it be academic for the content or administratively. You can ask questions of your peers as well, before, after, during the lectures or the tutorials, and it's really a good way to consolidate material if you ask each other questions. As I mentioned again in the intro lecture, there is the discussion board on Quirkus, and that is also available for your questions. We also have 2 outstanding TAs who are available for questions during your tutorial sessions. And again, there's lots of resources. As I stated, it's a big class, but definitely I'm going to try my best to make all of these resources available to you to the best of my ability. Okay. And again, as I mentioned, if you're interested, here's a great research article by Pam Mueller and Daniel Oppenheimer on actually how taking notes with a pen is more efficient than actually taking notes with a laptop. So have a read about that. Cornell also has some strategies, as does U of T. So you can also search on the U of T website for taking notes and the strategies that are involved with that. Okay, so what are we going to cover today? So what I'm going to first do is start just kind of to get you oriented with the idea that DNA replication is extremely important. I'm going to describe how the enzyme that replicates DNA polymerase, was first identified and purified. I'm going to then describe the biochemical reactions that DNA polymerase carries out. And then I'm going to describe some experiments that demonstrated some of the important in vivo properties of DNA replication. So you can kind of roughly divide this into an in vitro sort of approach or approaches and some in vivo approaches. And the reason that I like to show both in vitro and in vivo approaches is that the two complement each other in many meaningful ways. One way you can think about it is that you know the highly controlled environment of an in vitro reaction. One, of course, is able to manipulate the concentrations of the various components, and you can, of course, leverage that to find out many interesting things. But just because a protein does something in vitro, remember, doesn't necessarily mean that it does it in vivo. And we'll see that sort of towards the end of the first hour of this lecture, in that DNA polymerase, when it was purified, it was presumed to be the DNA polymerase that replicates the E. coli chromosome. It turns out that, in fact, the polymerase serves other functions. So even though it could replicate DNA in vitro, it was not the replicative enzyme. In vivo, you could also sort of flip the argument that if you have an enzyme that is doing something in a cellular context extremely efficiently, yet in the vitro context it doesn't seem to be working that great, that might be some indication that some condition of your in vitro reaction is not correct or optimal, or perhaps there are other factors that are involved that collaborate with that particular protein in the in vivo setting that you're missing in the in vitro setting. And then that then gives you some impetus to try to find and figure out and discover what you're missing. All right, so as you can see, I've inserted a blank slide here. And what I want to do here is actually just illustrate to you the scope of the problem when we start talking about DNA replication. So how many base pairs in the human genome? So you probably already heard that there are 3,000 mega base pairs, and that equates to 3 times 10 to the 9 base pairs in the human genome. Okay. The next question I'll pose is how many base pairs in one helical twist of the B form of DNA? So if you think back to when we look at the structure of DNA, you'll remember that there's about 10 base pairs per turn, and that equates to 3 nanometers per twist. And then the next thing I guess I'll ask is, are we haploid or are we diploid? So we are diploid. So that means we have 2 copies per cell of the genome. And if you actually do the math, that actually equates to 2 meters of DNA per cell. Okay, so the next question is how long do we live? So probably on average, we live approximately 80 years. I guess it's an average lifespan. So that's about 960 months. And then I'll ask, this one is probably a bit more challenging, what is the average lifetime of a cell in the human body? This one's probably a bit made up, but on average, the cell lifespan is approximately 2 months. So that means you have about 480 cell replacements or divisions occurring. Or in other words, cell replacements in an average lifetime. Of course, before a cell can divide, what does it have to do? Given that we're talking about DNA replication, it really has to replicate its DNA. So how many cells are in a human? Lots, right? So maybe around 10 to the 13 cells times 2 meters of DNA per cell times, say, like 500 divisions. And this would occur during a human lifespan, right? So that approximately equates to about 10 to the 16 meters. So what is 10 to the 16 meters? Does anybody know? It's actually a light year. So a light year of DNA. So a light year of DNA during a lifetime, so that is pretty substantial, right? And that's a big problem. You've got to make a lot of DNA, and you have to do it in a tightly regulated fashion, and you have to do it with high fidelity, right? I mean, it would kind of be irrelevant to call DNA the blueprint of life if we made really bad copies of it all the time. So basically, we're making a lot of DNA. We have to do it with high efficiency, and we have to do it with high fidelity. And of course, there's other reasons that are kind of, I think, important in terms of understanding and studying DNA replication. There are very deep connections between replication and human disease. So again, the key thing here is the connection to replication and human disease. Right? So obviously, if you fail to replicate your DNA with high fidelity, you can introduce mutations. You can introduce different kind of arrangements of sequences, gain or loss of sequences. These are all, of course, hallmarks of cancer. So here's an example of how mutations can cause cancer. There's very deep causal connections between defects and replication and repair in cancer. There's also quite deep connections to a variety of diseases that involve repeat expansions, and one of them is Huntington's disease. So you can have repeat expansions, and they include Huntington's. Another interesting thing of interest with respect to DNA replication that makes its study quite relevant is that it's a common therapeutic target. So again, that's another key thing, a common therapeutic target. We already talked about one. We have 5-fluorouracil. So 5-fluorouracil, which is, of course, an inhibitor of the thymidylate synthase. Sorry. So basically, it's a target of DNA replication. Okay, So if you're inhibiting an enzyme that is producing a central precursor for DNA replication, you know the real target for 5-fluorouracil is the DNA replication process. So again, it's an interesting process, and it's very important on a cellular level. And those are those connections to human health and disease. Okay, so where to start? So a pretty good place to start if we're discussing DNA replication is this. I'm sure you're all extremely familiar with this model illustration, which is from Watson and Crick's 1953 description of a proposed structure for DNA. And they managed to figure out, just by looking at this, that you could see how this might serve as a means of duplicating itself. And so that we have this image prior to the duplication. The hydrogen bonds are broken, the chains unwind and separate, and then each chain is a template for synthesis of a new daughter chain, and that's what's diagrammed here. Okay, so they unwind, and then we can get synthesis of a new daughter chain. The hydrogen bonds of the double helix have been disrupted, and a couple of new chains are laid down. So this model is pretty elegant. It's pretty simple, which of course is always nice. Simpler is usually better, but it doesn't really begin to describe how this process could happen. For example, are these chains just going to fall apart? So how do you, again, here's another question, how do you disrupt the hydrogen bonding and the other energetically favorable stuff that keeps the two DNA chains annealed to each other? Watson Crick pointed out that the two DNA chains are anti-parallel to each other, going in opposite directions. So the question became, how do you copy them? Does synthesis go in two different directions? So that's one question. Or do you have something weird going on that's in the same direction? When these chains are separated, are both strands copied at the same time? Is there the coordination between the synthesis of this new strand and the synthesis of this one over here? So do these happen at the same time? I mean, these are questions that they had very little knowledge of at the time. So obviously there's a lot of questions about how this process actually occurred, and the model just provided really just a beginning framework for how this might happen. So key questions: how does this happen? And you really needed to use this model as a framework for figuring out how, what enzymes would be needed to catalyze this process. So what enzymes? And what kind of substrates those enzymes used. So substrates. So once this model was proposed, there was, of course, even before it was proposed, there was quite a lot of interest among biochemists to identify the relevant enzyme or enzymes to start to characterize this process. All right, so the characterization of the enzyme responsible for DNA replication really began almost immediately. So the purification of the first DNA polymerase that was purified, which was done by Arthur Kornberg, Bob Lehman, Maurice Bestman, and Ernie Sims, as you can see, was done in 1956. So basically they went from the model in 1953 to characterization in 1956. So that was pretty quick. It was pretty quick progress considering all the inefficient tools that they had at their disposal at the time. They really didn't have all the nice techniques and equipment that we do now. So Arthur Kornberg, and he's shown here in this figure at the top, was very much a hardcore biochemist. He really believed in the power of in vitro reactions to eliminate enzymatic reactions, and perhaps even to a fault. So, you know, he points this out well, that this is probably demonstrably false. But at any rate, you know, he points out that without the cellular constraints, you can really push reactions to do what you want them to do by manipulating substrate and enzyme concentrations. And of course, this is true. But it's also possible that one can manipulate an in vitro setting such that it no longer even resembles the in vivo setting. So I do think some caution is warranted, and this is why I like the idea of looking at both in vitro and in vivo assays to really figure out how things work in the biological setting. Okay, so the idea is pretty straightforward here. And the idea is that if you have an assay by which you can detect the activity that you want, and you have some sort of you know, source of that activity that you're interested in, you can actually combine the two to purify the protein that's responsible for the activity. So basically, if you have an assay that you develop to detect the activity that you want, and you have a source of the activity, you could actually purify your protein that's responsible for the activity. Okay, so what's the activity that we're actually after here? We want to polymerize DNA, right? So we want to really start with some kind of precursor, and we want to make a polymer of DNA. And then once we've achieved that, we probably will, since we're polymerizing DNA, give it some clever name like DNA polymerase, for example. So we need an assay, and what does Kornberg actually have in his assay? Well, the first thing he has is thymidine, which is depicted here. And why does he have thymidine? There's at least two reasons here. So think about why thymidine. Well, the major one is it's a precursor for DNA. And it's exclusive to DNA, right? So we're talking about DNA replication here. We're not mentioning anything about RNA, so we really want something that's going to be specific for the synthesis of DNA polymers. He's also included ATP and magnesium. So why do you imagine he included those? So let's first start off with why ATP. Well, he assumed or presumed that because they were building something here, he would actually have an energy requirement. So energy requirement. Because we are talking about an anabolic process here. So they kind of guessed that they would need it, so they added ATP. Magnesium, they added it because it's a divalent cation, and that enzymes involved in these different kinds of nucleic acid metabolism reactions, they tend to use ATP, and also have magnesium around. So they just added that because they also thought it would be a good idea. So again, this would be needed for the enzyme requirement. those that use ATP also like to have magnesium around. And then they have their source of activity, which is a bunch of E. coli cells that they ground up and produced an extract. So how come they used E. coli cells as their source? I mean, why didn't they, for example, use differentiated human neurons? Well, the thing was that they wanted something that was undergoing active DNA replication. So they wanted something that makes a lot of DNA. And because E. coli cells make a lot of DNA, they're probably going to have the enzyme or enzymes present that are required for making a lot of DNA. So this is our source of activity. And why did they use E. coli cells? It's actively undergoing replication. So that was one reason. Of course, E. coli also affords other advantages. You can grow a lot of it. So again, grow a lot. So if you're anticipating that maybe there won't be a lot of this particular enzyme per cell, it can help to have more cells. It's also cheap. And at the time, and of course extending to today, it was a very, very common tool that people in biochemistry and molecular biology labs were using. So it was readily available. So it was a common tool. And also, just as an aside, if you're a human, it really hurts to have your differentiated neurons removed. Okay, so the last thing they needed was they needed some way of figuring out how they could determine whether they've made DNA or not. So how to detect their DNA polymer. So remember, they're starting with a thymidine precursor, and they wanted to end up with a DNA polymer, but they needed to have some way of telling the difference between the two. So key number one thing is that they used a radio-labeled form of thymidine, and they did this on this carbon here, where they used carbon 14. Okay, so they radio-labeled. So that actually allowed them to follow the synthesis of the DNA polymer by counting radioactivity. And then they need some means of telling whether this thymidine had been incorporated into the polymers of DNA, and what they used was a difference in physical properties between the thymidine and a DNA polymer. And the difference they chose to use was based on acid solubility. So if you add a strong organic acid to a solution of nucleosides and nucleotides, so these two, they remain soluble. Okay. Whereas if you add the same organic acid to a solution of a DNA polymer, so polymer, they become insoluble. And very simply, you can separate soluble from insoluble by centrifugation. So the idea would be they would combine all their stuff together, add some acid, and they use trichloroacetic acid here. And then this mixture would have a bit of cloudiness associated with it due to some things precipitating out. And then they would centrifuge. And now they have, the pellet of our insoluble stuff on the bottom and a clear supernatant. So you can remove the supernatant, count the radioactivity that's recovered in the pellet, so count the radioactivity, and if you've made a DNA polymer, the expectation is that you should have some radioactivity in your pellet. So that kind of makes sense. Okay. So they did this, and then they discovered there was some good news and there was some bad news. Okay, so the good news was that they indeed did see some insoluble radioactivity, which suggested that they made DNA. So there was some carbon-14 detected in this acid insoluble fraction. The bad news is that it was a very inefficient reaction, because what they saw was about 0.01% of the input radioactivity was converted to this insoluble form. So